Of all the molecules that we know on Earth, proteins are thought to be the most structurally sophisticated ones. I mean, look at it. It has bends and folds and curves that a lot of molecules don't. And it's very, very big. And in fact, it's so important in life that it makes up over 50% of the dry weight of cells. So if you remove all of the water within cells, proteins occupy most of what exists within the cells. Our bodies produce proteins, and all organisms produce proteins via DNA. And we'll go through those processes later, but the main functions of proteins are structural support. In our examples, in our bodies, we actually use fingernails and bones. There's storage support, such as in seeds. Plants get energy from the sun, and then they store that energy in proteins and in the form of seeds. That's why it's so important for vegetarians to eat a lot of seeds, because that's really the only form of protein that exists within plants in any abundance. Transport. Hemoglobin is a protein in our body that takes oxygen in our lungs and goes through the blood system and provides the rest of our body with that oxygen. And it is really good at binding to oxygen and releasing it at given points. Proteins are important in movement. So in muscles, our muscles are pretty much all protein, and actin and myosin interact, and they are actually what cause the flexing and relaxing of muscles. And so muscles are basically all protein. Also, proteins are really important in defense against foreign bodies, diseases, viruses. Whenever you get sick, your body produces what are called antibodies, and these are specific proteins that are developed with a specific structure that can help you defend your body. Also, proteins are important in speeding up reactions, and anything that is known to speed up a reaction is known as an enzyme. We know that there are hundreds of thousands of types of proteins that exist within the world. And really, it's their three-dimensional shape that defines what they do. In other words, the structure of a protein determines what its function is. And as you can see, there's several different structures of proteins. These are all examples. Some are nicely shaped, like this hexagon that looks like a snowflake. Others are more randomly shaped. But all of these have a very specific shape which determines what it does.
Amino acids are the building blocks that join up to form larger, complex molecules called proteins, the worker molecules inside all cells. Some proteins you may have heard of. The hemoglobin found inside our blood cells is a protein. Its job is to pick up oxygen molecules and carry them round our body. Antibodies are proteins. They're dispatched by our immune cells to attack foreign invaders. And hair and nails get their structure from keratin. That's a protein too. There are literally thousands of weird and wonderful types of protein, frantically busy carrying out all the activities that make cells alive. And all of these different proteins can be made by combining just 20 different amino acids.
Wow, that was a really cool video. So let's look at what proteins are made of. Well, proteins are hierarchically organized. Remember back to the very first slide I showed at the beginning of this class, and I said that biology is all hierarchically organized, and proteins are no exception. All proteins are made up of atoms at their basic level. Those atoms form into molecules called amino acids. Now, you can think of amino acids like letters are to words. Amino acids are the letters, and the words are what we think of as polypeptide chains. And so just as words are combinations of letters, polypeptide chains are combinations of amino acids. And just like words don't make up a whole story, polypeptide chains, which are like the words, are combined into big units called proteins. So we can think of proteins like a whole story, the polypeptide chain is the words, and the amino acids as the individual letters. So amino acids are the fundamental building blocks of proteins. all proteins are made up of only 20 amino acids, and they're considered monomers. They're a single molecule that combines to make a polymer. And we're going to talk a little bit more about that in the next couple slides. These form into polypeptide chains. Remember, these are like the words. And polypeptide chains are polymers of amino acids. They're just groups of amino acids that are connected together in a chain. And it has many, many amino acids. And just like polypeptide chains are many amino acids, are polymers, proteins are many polypeptide chains that are combined together. And that they fold and coil in very specific ways. And we'll see that in the next few slides. All right, now it's time to get down to the nitty-gritty. All right, what exactly are amino acids? Well, an amino acid is defined as having an amino group and a carboxylic acid group. And if you look at the amino acid structure picture in this slide right here, you can see both of those. An amino group is defined by the nitrogen. You may have heard of ammonia before to help clean your house. Well, ammonia is made up of nitrogen. And so you can always think of ammonia, nitrogen, amino, nitrogen. And carboxylic acid was where amino acids get the acid part of their name. And it's made up of a carbon double bonded to an oxygen and then also bonded to a hydroxide, which is an oxygen and hydrogen connected. There's a central carbon, which all of these are connected to, along with a hydrogen. And then there's an R, which is known as the side chain. It's also called an R group. That is the thing that changes. All amino acids have an amino group and a carboxylic acid group and a central carbon, which is connected to a hydrogen. The thing that is different in each one of them is the R group. And there's 20 different types of R groups. And these R groups determine the properties of each amino acid. So here are those 20 different amino acids. Now, they have the same amino group. They all have the same carboxylic group. And they're all combined with a central carbon. Those are shown in the pink. The R groups are all the different ones, and those are the ones that actually define the different types of amino acids. Now, these are all different in the size, the shape, and how they interact with water. There are two different ways they interact with water. They're either hydrophobic or nonpolar, and what that means is it literally translates to water hating. And whenever these molecules or the R groups come into contact with water, they bend in on themselves and go towards the interior of the protein. There are also polar or hydrophilic amino acids, and this literally translates to water loving. And so whenever these come into contact, they actually point themselves to the water. So imagine you have a group of amino acids together, some that are hydrophobic and some that are hydrophilic. The ones that are hydrophobic or water hating are all pointing towards the interior of the molecule, whereas the ones that are hydrophilic are pointing to the exterior of the molecule. Now, there's also differences in pH, and those affect some other characteristics within biology. And some are identified as being basic, some are identified as being acidic, and a couple of them are classified as being neutral. Here we have two amino acids, and each one has a carboxylic acid group, a carbon with 2 oxygen connected to it, and an amino group with a nitrogen and 3 hydrogen connected to it, both attached to a central carbon, each with a hydrogen, and an R group. Remember, the R groups are the ones that make different amino acids. And they bind by a peptide bond. And a peptide bond occurs when 2 of the hydrogen and 1 of the oxygen break off and form water, H2O. And then a peptide bond forms between the 2 amino acid groups, forming a polypeptide. Now remember, poly means many, and peptides is, well, many peptide bonds. And amino acids group together in a chain through a process called polymerization to form polypeptides. And it's important to note that one end has an amino group, which gives it a positive charge, and the other end has a free carboxyl group, giving it a negative charge. So in other words, a polypeptide has a direction. So you might be wondering what the difference is between a polypeptide chain and a protein. They're not the same thing. Proteins are considered to be groups of polypeptides, and just like a yarn doesn't make a sweater, a yarn is a chain of fibers that are wound together. Polypeptide chain is like the yarn, whereas the amino acids are like the fibers. And the sweater has a specific three-dimensional shape with which we use to keep ourselves warm. So just like a yarn is not a sweater, proteins are groups of polypeptides that are precisely twisted, folded, and coiled into specific three-dimensional shapes. And that three-dimensional shape specifically determines how it binds to another molecule, and that determines its function. So peptide bonds form what's called the backbone of a polypeptide. And polypeptides have specific characteristics. They have an R group orientation, and so remember the R groups are really the things that determine the shape and function of specific proteins, and they do that by interacting with each other. The R groups interact with each other, or how they interact with water. They also have a directionality. They either have a nitrogen terminus, also called an N terminus, or a carbon terminus, or a C terminus. And the nitrogen terminus has a nitrogen connected to a carbon, and it's also connected to 3 hydrogens. There's no other place in the polypeptide chain where that is. On the other end is the carboxyl group, and it's also the only place on the polypeptide chain where a carbon connects to 2 oxygens. And so polypeptide chains have a direction. So here's an example of a polypeptide. You can see it has an amino group, it has a backbone, and then it is combined by polypeptides. Polypeptides flex because the groups on each side are connected by a single bond. That single bond really allows it to be able to move in any different direction, really 360 degrees around the bond itself. And so these polypeptides can twist and bend in any different shape that it needs to. Here's an example why the order of amino acids is really important. Sickle cell anemia is a very severe condition that affects humans. And in normal blood cells, they typically last around 120 days. But a person with sickle cell anemia, their blood cells only last about 10 days. And the only difference really between a person with normal blood cells and sickled cells is that there is one amino acid difference in the development of the protein of hemoglobin. And that produces a change in the cell structure that looks not like a donut shape like it's supposed to, but it looks like a sickle shape or a football shape. And the shape of that allows those cells to be much more inefficient. So a single chain in amino acid sequence can have significant effects on fitness. Proteins are organized in 4 different structures. In other words, they're hierarchically organized. The first structure is primary, and it's basically just the sequence of amino acid. What amino acid is attached to the next amino acid? And the effects of that can be seen in our example of sickle cell anemia that we just talked about. The secondary structure of proteins is its repetitive shape. And typically it forms 2 different kinds of repetitive shapes, either coils, which we call alpha helices, or folds, which we call beta sheets. The third structure is called tertiary, or tertiary structure, and it's combinations of these secondary structures, these coils and folds, but they have these irregular contortions between them, and that's caused from the bonding between the R groups and other hydrophobic interactions. The quaternary structure is the overall structure of the protein, and it's defined as having distinct subunits of these tertiary structures within it. All right, let's look a little more into secondary protein structure. Now, what causes it is these hydrogen bonds that form between peptide chains. And these bonds form regular bends in the structure of the amino acid sequence, forming coils, which we know as alpha helices, and folds, which are called beta sheets. This increases the overall stability of the molecule, and so it doesn't really like move around a whole lot. And that's all caused by these hydrogen bonds. Now, while the secondary protein structure has really two main types, tertiary structures can be very diverse. We have two different examples of what the tertiary structure may look like in a protein. The one on the left is predominantly made up of these alpha helices, so there's several of these coils, and then you'll see an abrupt bend and then another type of coil. The example on the right are made up of predominantly beta sheets. There's a couple coils, but most of them are made up of these beta sheets where these significant bends, and then there will be another beta sheet. These bends form from the interaction among these R groups and the interactions between R groups and peptide bonds. This causes these backbones to bend, creating a very specific three-dimensional shape. Here we have an example of a complete protein, hemoglobin. It's a tetramer. It's a tetramer because it has 4 specific tertiary structures to it: alpha 1, alpha 2, beta 1, and beta 2. So the quaternary protein structure is the overall structure of it. So how are proteins made? They're made with 2 different processes. The first process is transcription. Everybody knows that DNA is important for defining who we are, but how? Well, DNA makes or codes for protein. The first process which proteins are made is called transcription. It's when DNA makes a copy of RNA called messenger RNA in the nucleus of the cell. And then that RNA, that messenger RNA, goes out of the nucleus through the nuclear pore into the endoplasmic reticulum, and it meets up with the ribosome. And then the process of translation occurs. A translation is basically taking the blueprint, which came from the DNA, as messenger RNA, and it's given to the factory, which is a ribosome. And that ribosome collects all the different things that it needs, and it puts together proteins in the order that they were ordered in. But don't worry too much about the details of this, because we're going to go over it in exhaustive detail in the future. Well, this has been the lecture on proteins. I hope you had fun.